Hello, everyone, and welcome to today's AAPS eChalk Talk. Today's eChalk Talk is hosted by dsm-firmenich. You may submit questions at any time by submitting them into the Q&A box at the bottom of the Zoom platform. All questions will be answered at the end of the program. It is my pleasure to introduce today's moderator, Athanasia Conley, Global Market Development Manager of Pharma at dsm-firmenich. Dr. Conley, the floor is yours.
Thank you. Good afternoon, everyone. Thank you all for being here, and thank you to AAPS for giving us the opportunity to present here. In the next 30 minutes, we will be taking you through some strategies that we have developed here at dsm-firmenich to overcome common challenges with formulating cannabinoids, and in particular, CBD, cannabidiol. I would like to, first of all, introduce you to our two speakers. Dr. Zdravka Misic, PhD, is our Innovation Project Manager here at dsm-firmenich, working on CBD, and she's leveraging significant expertise and experience from her work in application formulation in the pharmaceutical industry. Dr. Christiane Schweigert is our Senior Scientist and Team Lead in the Formulation Material Science team, and she has considerable expertise with formulating CBD. But before I hand over to my colleagues, I would like to briefly introduce to you dsm-firmenich. We are a 30,000 people strong company that is dealing with health, nutrition, and care, and we have a history of about 100 years. In addition to being one of the leading vitamin supplier in the world, dsm-firmenich is steeply grounded in science because of our 700 million investment in R&D every year, but also our more than 2,000 scientists spread across 15 facilities around the world. So in cannabinoids in particular, our unique offering combines three elements. First of all, the highest quality cannabinoid ingredients. Secondly, solutions customized to your needs for solubility, stability, bioavailability, and taste enhancement. And finally, expert services to support you every step of the way in your R&D journey. So this is the vision that we have, that through our innovation platform, we are able to help you bring to patients value-added and patient-centric treatment options. But how do we deliver against that ambition and vision? I will hand over to my colleague, Zdravka, to take you through that.
Thank you, Athanasia. I will start first with explaining a little bit about CBD and the therapeutic areas that it is currently used in. So what we have seen is there are currently registered more than 200, so more than 220 trials where CBD is used as API. It has been quite intensive interest seen in science and research in the last 5 years. For example, there's more than plenty of publications in scientific field exploring and evaluating CBD for various indications. In those clinical trials, we have noticed that there are 24 indications for which CBD may have therapeutic benefits. And to point out, for example, there's a strong interest in pain management. There's around 44% of all the trials that are focusing on this in this area. Now about the indications you see on the right-hand side of this slide that have some already clinical evidence that CBD can be used for. For example, there is high evidence for CBD in refractory epilepsy. There is also a lot of evidence for Lennox-Gastaut syndrome, Dravet syndrome, and tuberous sclerosis complex. Namely, these indications are covered by the drug, that the only drug that is currently on the market, and that is oil solution Epidiolex. Now, there is moderate clinical evidence for some other indications, for example, schizophrenia and psychosis, and also anxiety, mood, and sleep disorders. There is not a lot of clinical evidence for the pain, as I mentioned, as well as for Parkinson's disease, dermatology, but for example, pain is really coming because the clinical evidence is, as we speak, gathered. Now, on the next slide, I would like to a little bit give an introduction about the CBD and what it can mean. For example, there is a hemp seed oil which contains little to no CBD or THC. Then there are extracts that are derived from whole hemp plant. They can be full spectrum CBD, which contain other cannabinoids and terpenes, also the THC, which is less than 0.3%. There can be also broad spectrum CBD, which does not contain THC, only CBD and other cannabinoids. But what we are focusing today and what is in usually for the clinical development used is the CBD isolate. So this is the product that is actually 99.9% pure. It does not contain THC or any other cannabinoids. So on the next slide, I would like to point out some qualities of the CBD API that, for example, we are using in our research. And I'm sure that many of you know, especially if you're coming from the formulation field, how important the quality, the impurity profile is of your active drug substance. Because during the formulation, this is something you are already bringing into the formulation. And of course, it is of utmost importance to have it at really high quality. So our CBD API that we are using is THC-free. It is naturally derived. It has highest purity of the CBD. And there is no batch-to-batch variations. What is important, of course, to mention is that it has been manufactured under the pharma GMP. So it is certified according to ICH Q7 guideline. It also has a very important licenses and GMP certificates. But when it comes to the regulatory dossiers that we have, that we bring in together with our CBD API, that's a huge list. You see here we have a drug master file, also active substance master file for Europe. And what is very important, I'm sure many of you know who work in this research, there is a monograph within EP that was published recently and will be from 1st of July this year valid. So all the CBD APIs will have to comply to it, and our active substance is complying to it. So this is the material that we are using in our formulation development. And on the next slide, I would like to start in a little bit and explain why we have embarked in this journey that has been more than 2 years now ongoing. We had really a mission to help patients. Namely, as I have mentioned before, there is a high clinical evidence for CBD currently on the and Epidiolex is the only product that is on the market. Epidiolex is oil solution. It is something that is a little bit challenging for patients when it comes to administration. Depending on the dose, they need to administer quite high quantities of the oil. So therefore, we really had a patient in our mind. We wanted to have something easier for their administration, patient-friendly. So we opted for developing solid dosage form, which will at the end, for the drug manufacturers worldwide who we use it, and for the other indications, allow them to make various tablets and capsules or different solid delivery systems. We have aimed also at higher API loading. Namely, the higher we have the loading in our drug product intermediate, the higher, the smaller will be the tablet or capsule that the patients need to administer. So therefore, it is also easier for them in the continuous treatments they are having. We also opted for optimal bioavailability. So by that, I mean enhanced bioavailability compared to the CBD crystals, so the CBD drug molecule, which, as I'm mentioning, many of you know if you work in this field, is a challenging molecule. It's a drug substance that has certain properties that are not easy to formulate and work with. In specific, due to those properties, the bioavailability of this molecule is low. And I'll hand over now to Christiane to tell you more about how we tackled those challenges of the CBD drug substance and what we have achieved.
Thank you, Zdravka, and good afternoon, everyone. And in the following, I would like to give you some insights how we approach the challenges of bringing crystalline CBD, so as depicted in the bottom right, into a solid oral dosage form and while increasing its bioavailability. As Zdravko already mentioned, the oral bioavailability is rather low, only around 6% of the crystalline material, due to its incomplete absorption in the gut and due to its pre-systemic elimination in the liver. The CBD is classified as class 2B drug with a solubility, limited permeability through the GI tract membrane, and incomplete absorption in the gut is due to its lipophilicity of a log P of 6.3. Its absorption can be increased in the presence of fatty foods. However, this might lead to high variability between the patients. And LCT, so long-chain triglycerides, can favor the lymphatic CBD containing chylomicron formation and an uptake, and thus the bioavailability compared to CBD dissolved in medium-chain triglycerides. However, what we have seen in our research, CBD solubility is limited in many different types of oils. When it comes to patient acceptance, the load and the applicability are important, as Zdravko mentioned. And when it comes to drug approval, the physicochemical stability is very important. So CBD is prone to oxidation and under acidic or alkaline conditions and with light. It may form pharmacologically active metabolites, and CBD crystals can crystallize in different polymorphs, which might reduce its bioavailability. Coming to commercialization of the product, the scalability of the chosen process to formulate solid oral dosage forms is very important to bring the product on the market. So we see here quite some challenges, and on the next slide, you see how we tackle those challenges. We screened many different technologies to get the best out of the CBD. So I start from left, go to the right. We initiated a work stream based on CBD emulsification and spray drying. And because we have in-depth know-how here at dsm-firmenich and with external partners, we initiated work streams based on hot melt extrusion, a work stream based on liposomes and lipid nanoparticles, as well as one based on spray drying of amorphous dispersions. Out of those 4 technologies, we generated more than 200 prototypes, and we had to narrow down the number of prototypes to find the best one for the clinical study. So we did a lot of physicochemical characterization. We measured the particle size distribution of the interface, for example, emulsions and of the powder. We did the solution tests. We characterized the solid state using XRD and DSC. We tested morphology and so on, and we initiated storage stability tests to also monitor the long-term stability of the prototypes. We further did bioaccessibility and bioavailability tests using CACO2 cell models and animal studies to find a most potent prototype that will go into the clinical study soon. The outcome of this very extensive screening phase was that we continued, that we agreed to continue with a prototype coming from workstream 1, so the emulsification and spray drying workstream. And when we go to the next slide, I would like to directly focus on the illustration on the right-hand side. This kind of formulation principle had some advantages over the other prototypes coming from the different work streams as compared to the crystalline CBD and as compared to the liquid oil dissolved CBD. For example, we were able to increase the CBD load up to 30 to 40%. Because we found a, let's say, non-conventional oil where we were able to dissolve up to 80% CBD in 20% of that oil. Our form is also readily dispersible in water and can be used in various dosage forms. And in the following slides, I would give you some insights into the bioavailability data and stability data we generated so far. It is interim data as our studies are still ongoing. So let's start with the solid state characterization. Here depicted is DSC thermograms of a 40% CBD spray-dried powder stored at 5, 25, and 40 degrees for more than 9 months now. What we see here are very flat curves, very stable formulations. There's only a small bump visible at around 65 degrees for the form stored at 25 and 40 degrees. And this little bump is attributed to the melting peak of CBD. And if we calculate this back into amount crystals, we end up with around 0.5% CBD crystals in a 40% form, so in a very high potent form at very harsh storage conditions. Using XRPD, we couldn't detect crystals, and based on the HPLC assay, we have seen a very stable CBD content as well. So our design principle and our formulation seems to be very stable. There are hardly any crystals present, and this is a prerequisite for the efficient absorption in the gastrointestinal tract. Coming to the next slide, which is the particle size distribution on the graph on the left-hand side of the inner phase, so of the emulsion. Using dynamic light scattering, we have measured a particle size of around 220 nanometer hydrodynamic diameter, and the particle size distribution of the spray-dried powder has a particle size distribution of around 30 microns SD50. So what you see here is a very narrow and reproducible particle size distribution. And this is important to improve the intestinal permeability and also improves liver bypassing, and thus also increases CBD bioavailability. The data you see here was generated during a production scale-up batch under production conditions, and we yield here, the yield was here 10 kg SD powder. So this is really a batch coming from production scale. So if we move on to the bioavailability data, on the left-hand side, the in vitro uptake is illustrated into the cells and into the basolateral side using CACO2 cell models. And the yellow bar depicts our nano-emulsified spray-dried form, and it performs comparable to the CBD fully dissolved in DMSO. The same applies to the graph on the right side. Here's the animal data shown, so the PK evaluation. Also here, our nano-emulsified SD form, also the bar on yellow, shows similar AUC values as compared to CBD oil, the commercially available benchmark product. What is remarkable here is that the variation between the rats was much smaller with our form compared to the reference oil. So on the next slide is some data on the stability on the HPLC assay. Our ongoing study shows that our form is very robust. So this stability study we conduct at 5, 25, and 40 degrees for now more than 9 months now. We see a very stable CBD content, and more important, all known and unknown impurities in our formulated product are detected at a very low level only. And this is very important for the drug approval and complies with ICH Q3B standards. To summarize again, our currently available data set indicates that our design principle, our formulation, and also our production process is very robust and yields a very stable form. And this is crucial for maximizing the therapeutic benefits of CBD across various delivery formats. And with this, I would like to hand over to Strafka again.
Thank you, Christiane. So I'll just say a few final remarks and mention that what's coming next for us this year, still this year and very soon. So we are starting a human clinical trial. So it's pharmacokinetic study where we want to show, to see the comparison with golden standard that is on the market. This study is very designed as very robust and with many participants. So we are quite sure and we are eager to find out how the results will turn out, because we are confident that the outcome will show us good, good results, and it will be it's very, very sizable. So we will be able to trust the data that we are getting. Also, after that, we are ready to go for commercialization. So as mentioned earlier, our product will be possible to be used in different delivery systems, oral delivery systems. So you can imagine various tablets, film-coated tablets, also then chewables, orally dispersed ones for different routes of administration. So here we are not talking only about oral administration. It's possible to go for some buccal or sublingual administration, of course, depending on the dose and indication that you would like to tackle. Today, we would also like to mention the opportunity that we are very open for sampling of our materials. So if you have heard from this, our presentation, we have two products that we could sample. One is the CBD API isolate, 99.9% CBD and very, very pure and with highest quality, as mentioned earlier. That can be used for oil-based drug delivery systems. And we have the formulated CBD powder, spray-dried powder that Christiane has now extensively described the properties that then can be used for the various solid delivery systems. Feel free to reach out to us, to submit your requests. We are very open to get your contacts and to evaluate potential collaborations. I believe on the next slide, we have some references to which prove our scientific contributions and evaluations. And of course, it's not just three of us. There's a huge team dedicated to this CBD drug product development activities, and we are happy to work all together to bring this forward and soon to the market. I think now we are open for questions. Feel free to put them in a chat. Also mention your name and affiliation, and we'll do our best to answer them. Of course, in case we do not have time for all of your questions, have seen many of them popping up already, we will come back to you later. So make sure that everything is answered. Thank you all.
Thank you very much, Zdravka and Christiane. So you can put in your questions in the tab called Q&A, and we will try to answer as many as we can in the coming 5-10 minutes. So one of the first questions that came up, and let me stop sharing my screen here. You can put the questions in the Q&A. So one of the first questions that came up, and that one is for you, Zdravka, is which applications can this technology be used for, and what could we learn from that?
Yeah, thank you for this question, and we are really happy to have this drug product intermediate in development, because it will allow many different pathways in designing the final dosage forms that the drug manufacturers are putting on the market. Namely, depending on the indication, there will be a certain dosage strength needed to be put in a tablet, let's say. So what we are bringing in is highly loaded powder Therefore, the tablet size will be quite small. Bear in mind that opens also doors for other actives. For example, if you're not going into the indications that only CBD is needed, there's room for other APIs. There is a room for other also excipients that might even change your release profiles. So yes, our powder is immediate release powder, but it can be, of course, I'm sure the many formulators here know, can be designed as a sustained release, for example, dosage form. So there's a different options from the route of administration, as I mentioned, oral, buccal, sublingual, then to different doses, different types of tablets, capsules, and then variety of the delivery systems.
Thank you. We do have another question on the formulation technology from Andrew. So Christiane, the question is, can you explain more about the formulation technology? How is it prepared and what is the composition?
Thank you for the question. So what we use for our formulation design principle is an emulsification approach. We emulsify CBD in an oil and stabilize it with a matrix. And then later on, we spray dry this foam. I cannot deep dive into the composition yet because we filed a patent application, but it is a conventional or emulsification approach and later on spray drying.
Thank you. So we do got another question from Deipansu. What is the role of cannabinoids in cancer treatment? So it's a very good question, and actually it requires a very long answer. What we can share today in a couple of minutes is we have seen an approved medicine in symptoms related to cancer treatment and cancer pain, that is CBD and THC. And we also see several phase 2 clinical trials looking into specific rare cancer types. So I would say happy to have a longer conversation. Do reach out to us, but there's definitely promise there. We got another question that I would like to address to you, Christiane, and that is about the solubility aspect. Does this CBD powder dissolve in water?
That is, this powder does not dissolve in water, but it disperses in water. And yeah, it disperses readily, so a couple of minutes, and then it's finally dispersed in the aqueous media.
And there I would like to add up that it opens the doors for another final dosage form that I have forgot to mention, for example, and that's about effervescent tablets, for example. This is something that you could also design using our drug product intermediate, add certain excipients to make it effervescent, and then definitely it's also one of the ways of administering CBD. So quite an interesting also approach there.
Okay, thank you. We did get a question on, you mentioned data available on patient convenience and compliance and the treatments that are available right now. Can you say a little bit more? So I'd be happy to briefly take this one. We do know that the maximum recommended dose with the approved, let's take an example, approved medication on the market with CBD is about 20 to 25 kilos per 20 to 25 mg per kilo of body weight. So if we assume an adult of 75 kilos, then we're looking at 15 milliliters of oil. There are some acute cases where we have to look at about 10 ml for a child of oil. So that is not only has organoleptic difficulties, but it is also hard to take. Sometimes for some people it is prohibitive to take this amount of oil every day. And also there is patient feedback consistently that more convenient, compact, easier formats would help them stick to their treatment. So there's a 2024, it's just a few months ago, a study that found that prescription CBD It was discontinued in 23% of patients in the first 12 months of initiation, and that was a combination of factors such as time to response, potential side effects, and the fact that the treatment was not convenient enough for them to be compliant. I think we have time for one or two more questions. So I would like to take this one about the storage conditions what are the storage conditions for this formulation? Christiane, could you take this one, please?
Yes. So we did as shown, we did extensive storage stability tests at 5, 25, and 40 degrees. We haven't seen any crystals at all at 5 degrees storage, and only a minor peak at 25 and 40 degrees. So we recommend 5 degrees, but also 25 with the current data set would be a good one.
Perfect, thank you. So we will take one last question that I see here on our Q&A. The question is this available? What could we do with it? Can we have access? What is the development status? So first of all, let me recap. We are taking this formulated CBD into a pharmacokinetic phase 1 clinical trial. However, in parallel, this is available. So it can be supplied for commercial projects that we are very happy to help you with that. And in parallel, we are able to provide samples for non-for-profit research. And you will get the slides, and there is a link there to a landing page where you can ask for these. Feel free to reach out. We are very happy to help you in your projects, and we are very much looking forward to partner with you. So with that, I think we've hit our time, and we will probably close off this conversation. Thank you all for being here.